See that we have really distinguished speakers here today that will tell about different aspects of personalized prevention in different countries. And I guess in many of talks you will see that this is strongly related to genetics, but I think what is important that you will also see that it is not only about the genetics, and mostly it's about the use of this information for the prevention and for the personalized prevention. So, and our first speaker in this session will be online presentation from Stefania Boccia, and she is director of University Department of Health Sciences and Public Health Faculty of Medicine from Italy, and I'm really welcoming her to provide us insight in the information on personalized prevention. So the floor is yours.
Good morning, everybody. Thank you again, Professor Massaslayer, for the invitation and for the moderation. I'm not sure if you can hear me properly.
Yes, we can hear you.
Okay, perfect, thank you. So I will present in the 20 minutes that have been assigned to me, the main activities relating to this project that I coordinate since 2022, and the preliminary results towards the generation of a strategic research and innovation agenda in personalized prevention and the correlated roadmap. So, next slide. So the project is a coordinating and support action that is participated by 18 partners, and we are already at the third year of activities, whose goal is to co-create a personalized prevention roadmap and the strategic research agenda in order to support decision makers in the definition and implementation of personalized preventive strategies. It is one of the projects under the umbrella of the International Consortium of Personalized Medicine that brings together 50 participants and representatives of European member states from the academic, the ministries, and also the funding agency and the European Commission itself. So we had a kickoff meeting in Rome quite a long time ago, and at the time the project started, we worked a lot towards a common definition of personalized prevention that was not evident at the time. So the definition of personalized prevention that I report here, and that is addressed also in these academic papers, basically it's an approach that doesn't want to substitute the traditional way in which we make prevention today, but rather to integrate by leveraging on genomic data, on the large amount of data also from environmental factors, lifestyles, socioeconomic factors, all possible data that can be gathered and combined in order to stratify the population into segments of different risks, so that in principle, the traditional way in which we make prevention can be more precise and even more effective and efficient. Just as a professor of public health, I would like to remind you the relevance of prevention itself in keeping the population healthy, not only as long as possible, but also in terms of quality of life. So if you can look at the slides on the left part, you see that clinical medicine affects, this is a rough approximation, only 20% of the outcomes, while health behaviors contribute by around 30% of health outcomes, and also socioeconomic and physical environment factors. A nice report from McKinsey underlined that for every dollar spent on prevention, the revenues is more than double. So prevention is really a key in order to keep also health systems sustainable, and not also improving the quality of life of people, which is, of course, very important. If you look at the slide, in fact, you can see quite clearly that the length of life of women and men living with some disability in Europe is not as huge as expected. So on average, individuals in Europe live around 20 years in a bad quality of life. So the role of prevention is really key. We are not new, me myself, in working personalized prevention. We were pioneer in this actually field since 2014 with this Maris Lodowska project that released some recommendation in 2018 that I don't want to address today because I want to talk more about profit, but I just wanted to remind that already 2018 we released some recommendation on how to integrate personalized prevention into personalized medicine by pointing out the relevance of identification of biomarkers whose clinical efficacy is evident, but working on that economic evaluation, but also ethical, legal, and social aspects, and also organizational models, because we have a lack of competencies among health professionals in understanding and delivering results of predictive genome testing. I want also to say that personalized prevention falls also in the broader concept of precision public health, that for the first time has been coined this term from Stanford Medical Group in 2018, in the same year that we released our recommendation on personalized prevention. So I don't want to read the slide, just for you to remember that precision concept encompasses not only prevention, but also diagnosis and therapy. And the last year, there was a very nice report from his colleagues in the United States that underlined to what extent precision prevention concept has evolved recently by keeping attention. The methodology used has been enlarged because we have now AI and language learning model that can facilitate the uptake of the large amount of data available to make personalized prevention in the context of precision health be a reality. And also wanted to keep your attention to implementation science. So we don't need only evidence of clinical outcome improvement, which are of course important and from the basis, but also implementation science that look at the more correlated concept of organizational aspect, feasibility and acceptability from citizens and patients of these technologies. Luckily, the European Commission is investing a lot in implementation research in personalized prevention. As you can see in the slide, we made an overview of the amount of budget is more than 220 million invested in the past 3 years in implementation projects in personalized prevention. So I think we really are now at the beginning of a new era. So looking back at the profit of specific objectives, it is a support action of the European member states. So the aim is to deliver some concrete roadmap that can be used at the European member states level. So this is why all the objectives of profit that you can read in the slide has been developed through a strong collaboration with a number of stakeholders. Actually, some of the partners are also in this panel session. So I hope that they can testify how much we really will be working with a lot of stakeholders towards awareness to a large extent on the relevance of personalized prevention. And concerning the methodology that we have been using, we started with a large mapping exercise during the first year of the project, moving to assessment phase. And all these 2 phases, I will keep an eye now about that, we're feeding the strategic research and innovation agenda that I will talk at the last part of my lecture. So what we did in the mapping phase exercise of the Profit project, you can read more in detail if you scan the QR code, which were the activities and which were the major results of the mapping exercise, because we don't have time, it's 20 minutes to look in detail. So I'll just give you some flavor of what we learned from this mapping exercise in the next slides. So the first major mapping was trying to have a sort of prevalence picture of what was going on concerning the available personalized preventive approach for chronic diseases by looking at both primary studies, but also clinical guidelines, again, in the field of chronic disease prevention. But also we were looking at the bottlenecks that preclude their effective implementation. So the results of this scoping review revealed that the vast majority of available personalized preventive approach are in the field of cancer and are in the tertiary prevention. So we talk basically about pharmacogenomic, but also target therapy for cancer. But more recently, we see more research available in terms of primary study in primary prevention and secondary prevention. By looking instead at the reviews on the bottlenecks, they revealed that today we have more than 300 unique bottlenecks present that impede their effective implementation. We try to group in 6 major domains, and in these 6 domains, you can see that the bottlenecks are lack of clinical efficacy evidence, but also portability across population of some polygenic score, but also poor knowledge of health professional, poor understanding of the public and potential for discrimination that they advocate, but also healthy issues and economic issues. Concerning the consultation of experts, but also the public concerning the bottlenecks, I can say that these topics have been, as a bottlenecks, have been confirmed. But also the major cross-sectional bottlenecks is the poor awareness of the relevance of prevention and the amount of budget, unfortunately, spent on prevention today in the member states. We also made a large scoping review about what are the actual personalized preventive events or collaboration involvement that involve the public. There are very few actually present today. So I think there is a lot of space to improve our way we engage population in understanding personalized prevention. So very quickly, moving into the other phase of the project, the assessment phase, we were working to develop a new framework that can be beneficial at the European member states level to apprise personalized preventive approach before introducing in the healthcare system. So just if you look at this picture, it's given from Public Health Genomics Foundation, wants to mean that we do not need only analytical validity, but also clinical validity and all the other elements of personalized outcome, value information, feasibility of the test, and cost effectiveness when we talk about the clinical utility or whatever genetic and genomic, but other only predictive genetic testing in personalized prevention. So what we developed is not published yet, but we will do very soon, is to release a sort of holistic framework that takes into consideration not only the health technology assessment when it comes to introducing a personalized preventive intervention, but also health impact assessment. So what does it mean? It means that when it comes to introducing a personalized preventive intervention in the European member states, we should take into account the stakeholder perspective, the feasibility and acceptability of patients with bad general practice, the capability of healthcare professionals to do it, and the more general impact in improving outcomes, not only at the personal level, but at the population level, and also monitoring whether it works in the short and the long term. So we did test our product framework, practically speaking, in 3 different settings, the use of the PYD testing to prevent side effects, severe side effects, that although there is national policy that mandates the use of the test, unfortunately the use is not widespread, and we try to understand, using the product framework, which are the barriers that impede implementation. Also the BRCA genetic testing for family members of positive people. This is again in the recommendation, in the guidelines, but it's unfortunately not much implemented in the European member states. We try to understand which are the barriers that instead should be overcome to implement this test, and also the pharmacogenomic passport. This is really at the forefront of innovation, but using the health impact assessment framework, we try to understand which are the factors that today, at least in Italy, impede the implementation and should be addressed in order to use it as soon as possible, because actually the evidence of clinical efficacy are very strong from trials, and in fact, this is already used in the Netherlands. So, concerning the strategic research and innovation agenda, and I have to move basically to the end of my lecture, we already released the preliminary draft. Okay, so I cannot talk too much about that because we didn't deliver the final results to the European Commission. But what I can tell you are some hallmarks that we agreed upon as partnership, because we identified, based on all this work done that I tried to summarize in a few slides, we tried to identify 10 major challenges to prioritize in order to, for the member states to keep an eye and to make actions in order to cope the gaps that are present across all these 10 challenges that you can read by yourself. This fact sheet that report the main elements of the strategic research and innovation agenda can be downloaded. So if you want to have a copy of this fact sheet, you can use your cell phone for this QR code. And I just want to for you to see just one challenge, just as it looks like. So we have no time, unfortunately, to read this challenge, but for each challenge, we decide which are the gaps, which are the priorities for implementation. So we give guidance to cope this gap and what it really matters for each of the 10 challenges identified. So unfortunately, we don't have time to go in all these challenges, but just for you to see, just as an example, the broad scope of promotion and prevention. Why this is important? Because there is strong evidence that if we act on prevention, we can improve health outcome. However, there is weak integration with social determinants and prevention is underfunded. So we think that the priority is to integrate biological and other contextual factors and to invest more in prevention itself. And this is the same for all the 10 challenges of ZRIA that you can read in summary, because the final ZRIA, which is like 60-page documents, will be released, unfortunately, at the end of this month, so I cannot anticipate the final version today. But again, if you want to read more, you can read the fact sheet, which is a nice summary of the content of this RIA. It's up to the member states to keep this RIA seriously and to put something in practice to make it concrete. In the roadmap, there is the accompanying document of this RIA. Again, you can read the summary with this QR code. We identified very concrete action in the midterm, in the long term, and who has to fund and who has to lead the actions. So again, this is just a snapshot picture of one of the challenges where the roadmap of the action have been identified. I know that the European Commission is waiting for that, so unfortunately I cannot anticipate too much. Last slide, for you to know that we are working in strict connection also with a joint action, which is the network of cancer expertise, where we identify a network of expertise on personalized prevention. And of course, we work very strongly with Marcis Leia, Professor Leia, in the European joint action in cancer screening, where we work and lead a personalized prevention work package. So thank you very much for the attention. The final RIA will be presented at the European Public Health Congress, where also Professor Perola will participate and the partners of this nice project that I was presenting today. Thank you very much for your attention.
Okay, thank you very much for this really comprehensive presentation of these well-needed activities, because I think indeed one thing is to hear the different success stories, another is to really understand what are the real bottlenecks and everything to be that we need to really make this personalized prevention working. I'm reminding that we do not have the questions right after each presentation, but we will have the panel discussion after all presentations will be done. So with that, I move to the next presentation. The next speaker is Andres Metspalu, professor at the Tartu University, professor of Genomics and Biobanking. And I think many of you know Andres, and I think I can really say that he actually put Estonia, strongly put Estonia on the map of biobanking and genomic research. And I always know that if there is some mentioning of top leading biobanking activities, Estonia is often mentioned among the biggest names in the world. So basically, I think this is mainly through the activity of the Andres that has been done, and most importantly, that does not stop at the biobanking. So it's implemented in healthcare as well. So floor is yours, Andres.
Thank you, and good morning, everyone. So, what I'm going to talk today is why do we need a personal medicine? What is the problem? And, you know, Stefania now listed 10 issues, what are the problem. But bottom line is that the waiting lists are too long. It takes sometimes 6 months to get to see a specialist doctor, like ophthalmologist or orthopedic surgeon. There's not enough money. Centralized healthcare systems are running out. And we just have too many patients. That's the reason. We have too many patients, and because we are diagnosing better and so on. So, and the reason for this is that we are not putting enough emphasis on prevention of disease. Personalized prevention, that's why it's now the next big wave, just to transfer the money and people from treating disease to prevent disease. And this is what I'm going to talk a little. So that means biobanks were very good for this. Biobanks are kind of experimental thing. It's for research and science, but biobanks are not providing health care as such. They are preparing infrastructure, training people, and so on. So I'm going to talk things about the Estonian biobank and how we use the biobank for trying to transfer the knowledge into healthcare. Estonian biobank is governed by law, it's Human Gene Research Act, and the law at 3 points only: to collect information from Estonia, to analyze it, and to use the data to improve the public health. So now we are in the third phase. So I'm going to give some examples, and finally I give my vision of the future. Like, you know, it's really has been long way, you know, like we started 99, last century, previous century, you know. This is really interesting. We have 200,000+ people in the biobank, and pretty good data, updated regularly annual basis, and also lots of molecular data. We have, I mentioned, we have a law, and we have clear access rules also, so we are collaborating with many institutes, universities, other biobanks, and so on. So what we have, I wanted to really draw attention that we have in biobank, it's a voluntary biobank, and it was not a classical epidemiological thing. You send the letters and you know, get something. We did lots of public announcements, and you see more women joined the biobank because they felt that they are responsible for next generations. So when I was asked why did you join the biobank, you know, it was probably too late for me. I've been smoking already 25 years, but I have children and grandchildren, and maybe, you know, this science helps them. So based on data, you see we have a primary care diagnosis, like almost 30 million diagnoses. We have 25 million drug prescription data points. Questionnaire, over 200 questions. Pretty much classical genetic epidemiological questionnaire. And we have close to 40 million SNVs, which we can analyze and can use for imputation. And we have a secondary care diagnosis, like almost 500,000. So regarding to the molecular data, so we have, everybody has been genotyped with Illumina array. Everybody has NMR data on using Nightingale technique, which is a little more than 200 different small molecules and lipids and so on. Very useful. And now we are sequencing we have 5,000 people sequenced with short read, the Illumina technique. Now we are sequencing long read technique using PacBio technology. I have to say that this is really, good, very good data. You can see things which I haven't seen before. So I also have to be proud of it, because Nature Medicine published last year a review and listed 15 biobanks. You see our friends from Finland and UK biobank, who is really good. There is a tiny dot where is a Qatar. Qatar, they do very well, because they have to do it, because 40% of marriages are between cousins. And so they are running into genetics. And also Estonia is listed. Of course, US have lots of biobanks, but all of us is, I would say, is a major biobank. China, they can do everything. They have a Cadore biobank. 50% of samples are in Oxford, 50% in Shenzhen, and Japan biobank. So this is a very nice map, actually covers the world globe. Now Africa is also picking up. For instance, there is 100,000 individuals will be sequenced in Egypt, and South Africa is doing very well, but also Tanzania, Nigeria. So Africa is catching up. So what's the Estonian biobank story? It was published this year, April, Nature Communication. So everything was put into this paper, what we have been doing. Now the examples. I'm also a medical doctor, so I know them all. Basically the problem is that they always wanted to collect the phenotypes. Phenotypes, but genotype was the last thing they were thinking of. But we just thought that, and not only us, but this is now a common thing, we can start from genotyping, which is very cheap. You get genotype, and you already know what you have to look for. So we did a FH study, which is genotype first. We took only variants from 3 genes, like LDR, ApoB, and PCSK9, and elevated LDL-C levels. Not really big ones, just borderline, upper borderline. And we invited these people to cardiology hospital, and cardiologists were very skeptical at the beginning. Oh, we know these patients all. But what actually turns out is that they had only 3 patients, which, if you look on the left side, they had only 3 patients from this cohort they know. The 17 had no diagnosis whatsoever, and 21 had elevated LDL cholesterol. And the recommendation was, eat less, run more, it goes down. It will not go down like only by running. You have to do something else. So after visiting the cardiology hospital, look, 37 got statins immediately. Statins, saving lives. I know there are lots of people like anti-vaccines, there are also anti-statin people, including medical professionals. But still, this is what, as much as we know now, it's useful. So, so this is treatment goes. Of course, there are people who couldn't really take statins because they were either pregnant or were emigrated or we couldn't get them anymore. But still, it's very useful. Now, of course, cardiologists are really happy. They are starting their own registry and so on, so they want to get them all. This is called like a rare disease. It's 0.5% in the population, but it's 1 out of 200. But if you have now calculate, you have 1 out of 200 from 1,000, you have already 5. From 10,000, 1 million, 3 million, you know, it piles up. But these patients, they don't feel almost nothing. First time they see the doctor is an emergency unit, and quite a few not coming out of it. So it's important. Okay, this was specific genes, specific variants. Now there is a new technology which we call like everybody's called polygenic risk score. So this is the thing, you have a rare disease, one gene, one mutation like cystic fibrosis, more than 60% of patients have just one mutation if you take Huntington, all this stuff. But they are really bad diseases, but there are only few people. For instance, in Estonia, PKU patients, we just get 3 to 4 per year, and we screen all newborns, but still useful, still cost-effective. Now, almost 50% in our country, 46% last year died from cardiovascular disease. 23% are dying from cancer. So how can we implement genetics in these ones? And that's why we did the biobank. I started 25 years ago. So you see the standard, okay, you see the standard distribution. If you just calculate the polygenic risk score on the biobank people. And you know, I have to look at these ones. These are very high risk of these certain phenotypes. In order to do it, you have to have the data. You have to do GWAS studies, which is genome-wide association studies, impute array data. If you have to have a really good sequence panel. Now the Genome of Europe is producing 100,000 new genomes from 40 different populations across Europe, so we can use this one in a few years. And then, if you just transfer, oh my God, if you transfer this into this, you see that the top 1–2% are as important as monogenic variant. And you have to deal with these ones, the same, but it comes from population, and numbers are much higher. So now the question is, so we are all different, you know, Latvians, Estonians, you know, Lithuanians, Finns. Basically, all these countries around us have basically the same thing. In Europe, we can all use UK biobank data, more or less. But now Europe is not anymore Europe, so we get lots of people from, you know, Syria, everywhere, North Africa. So we have to be a little bit more open. And for certain populations, we can't really apply this polygenic risk score. Although IT guys are really calculating all kinds of tricks, but you can't really do it. You have to really measure the real life. This is what, if I would like to do medicine, someone has to make an action because of this. So you better have a best data available. Now, this is important slide. So it's a cardiovascular disease incidence. It's a 5-year thing. Now, what people are using, doctors are using now, they're using risk calculations. They're using so-called score method, which doesn't involve genetics. And this is here. Now, if you apply polygenic risk score on this group of people, then you see that some of them go into low risk group, you don't have to do anything. You just save money. Otherwise, all this group gets the same treatment, same attention, same approach. But these ones we can get out. Some of them are getting very high risk, and these guys need special treatment, action, immediate action. Otherwise, they just might be too late for them. And most of them stay like this. So this is the thing, and it's only 25 to 60, 59 years old. So now, if you take 60 plus, so basically it's the same thing. So this is how PRS actually works. So you just can stratify the existing data. So doctors like it much more because they are used to do with this one. Now they don't have to start from the zero and learn something. If MRI came, nobody had, the primary care level. They had no need to learn physics, what is the MRI. They just read that the radiologists are writing 2 lines and transmit this to the patient. Okay, but if you take top 2% and take this, let's say, 60 years here, 60-year-old. I'm much older, but 60 years old, you have a bottom 25%, about 10% of risk. Top 2%, 30%. 30% means that in this group, every third might get the event. You have to do something. You are 60 years old, and this is risk. You are really in real danger. So, and this is not coming out using normal procedures what hospitals are using today. So again, this approach is important. I would say PRS is the most innovative and novel approach in healthcare, population-based. It reminds me a little bit newborn screening. You just screen all the newborns, and your action is taken in cases what is really actionable and what you discover. And it is really cost-effective. It's the same for us. We are testing cars every year, paying ourselves, but nobody's testing the full population. So next thing, breast cancer. I don't see where is. Oh my, running out. So you see the breast cancer too fast. So breast cancer, you see, it's also, some people have higher risk, some are lower risk. But if you take a summary, you see, this is population risk, and this is top 5%. That means the same risk happens 10 years earlier. Instead of 50, instead of 70, it's 50. So it's again important, because now the screening starts at 50, and you get the letter. If letter is not, lots of women don't get this letter because they are already dead by this day. Again, if you take the top percent, it goes really high. So it's again important. Important is just to take the top risk at the beginning in order to get the most of the. Okay, I leave this out. And finally, I have Parkinson's disease. This is now important because Parkinson's is also, we can see the risk using polygenic risk score, but it's risk what to do next. And now there is also, I would recommend, my vision is that we have to use the next test in order to refine high-risk people who is really getting this disease. In cancer, we could do liquid biopsy, which probably gets the information 2-3 years before you get the imaging in MRI. In Parkinson, there's also imaging techniques, but there is one very simple and cheap thing, is a transcranial ultrasound analysis of substantia nigra. And we did the same thing. We invited 100 people with high risk and 100 with low risk, and this is the image. And you see that high risk, you get 9 people were immediately diagnosed disease. They were running around, they didn't know that they have a disease. And low risk group, you get 3. And if there is a certain cutoff, so almost 35% 36%, 46% were above the cutoff. That means these are people who are developing Parkinson next 10, 15 years. Useful to know because you can postpone a little bit the disease if you are active mentally, active physically. Okay, so it works, and it was published also. So I skip this one because it's already there here, and it's important how to transmit information. Challenges. Of course, there's lots of challenges because it's a new thing. You know, people, medical profession is very conservative. They have to really get everything tested many times. But there is a big thing is primary care and prevention and hospital care. Hospitals actually, honestly, they are not very much interested in prevention. We are all trained to treat disease. And it's important that we can treat disease. But there is enough patients, but we have to still do more prevention. Okay, so biobanks are useful just to model things and then transfer everything to population scale. But I would say the biobank concept is in a way already old. We have to see in modern times the full population as a biobank, a kind of biobank, because we have data on everyone and we can approach individually doing intervention. And I also moved from BRS to the next test. You know, almost every case you can have something. If you have risk, doctors tell me, look, risk, what I'm going to do is this risk, but I need a little bit more. So for this small group of people, we can apply a more specific test. And so my vision is that we have to, as a population, we have to sequence a certain number of people, do everybody else array, impute arrays, get PRS, calculate risks, and send this information to the primary care. Primary care has get this somebody on the screen, Andres, your glaucoma risk is very high. Just go to the ophthalmologist, probably say, do something about it. You don't lose your sight. Thank you very much.
Okay, thank you, Andres. Yeah, really, the concrete examples, I think that's something that we need to show the usability of the biobanking in prevention. Okay, now I actually give floor to you to chair the session.
Okay. So the next speaker is Professor Markus Perola. He's head of the FinnGen group in Helsinki THL Institute, Epidemiological Institute, and also has a research position in the University of Helsinki. He has been a long-time collaborator and also doing biobank for a while. He got a PhD from Helsinki University, did a postdoc in UCLA and on statistical, so he's very well also in statistical genetics. So please, Markus, floor is yours.
Thank you, Anders. Nice to be here again. I brought the Finnish weather with me. So I'm going to talk to you about the FinnGen-project update. I mentioned this already 2 years ago, but I presume most of you weren't here 2 years ago, and now I have a lot of updates. But I wanted to start with a description of the project for the ones who weren't here 2 years ago. I am, as Andres said, member of the FinnGen steering committee, head of the FinnGen in THL, which handles all the registry data and DNA logistics. The PI of FinnGen is Professor Arno Palotie from University of Helsinki, FIM, and he's really the father of this beautiful study. So what FinnGen is this, I will repeat myself in many of these slides, but in general, during the last, well, I should say actually last 20, 30 years, we have collected up to 500,000 Finns for different population cohorts, hospital-based biobanks, etc. And 2017, we decided to go for a GWAS genotyping and do a GWAS-FIVA study on these individuals. About 2/3 of the FinnGen participants have been collected since 2017, so we had already about 200,000 samples a little less than when we started this so-called legacy data. So the idea is to do genetic association analysis, as I said, and also to enrich the biobanks, contributing biobanks data. The thing is that unlike FinnGen is called in many places, FinnGen is not a biobank. FinnGen is a project, which consists of data from 11 different biobanks, but I don't mind. You can make call FinnGen a biobank, but anyway, legally that is what it is. So it is also a public-private project. A small country, we couldn't have money to do this kind of UK biobank scale thing, so what we decided to do from the beginning and what actually enabled this is to work together with pharma. So currently we have 14 different big pharma companies with us. This is a pre-competitive study, so the pharma scientists work with us, public scientists shoulder to shoulder, and the IP rights are not considered at the primary analysis, which is done by FinnGen analysis team. As on the left, you can see the biobanks involved in this. In Finland, we have a very good biobank law, which allows this kind of a study with very wide consent. And as I said, all the Finnish biobanks, registered biobanks, are currently part of FinnGen. So, as I said, we started in August 2017. Of course, the planning started many years before. Big part of that was the ethical evaluation. I'm actually responsible for ethical issues in this study, which is part of my role in the steering committee. The FinnGen 1 was like a trial period for 3 years, and after that the funders were very satisfied with the work we have done, and we got continuation to FinnGen 2 and now new funding for FinnGen 3 as well. I must say that a significant part of this is also funded by Finnish government, Business Finland. It's a government-based body supporting this kind of public-private collaboration. About 20 million euros came at the very beginning, which helped us to collect the samples, extract DNA, and provide that DNA and data for the project. So actually the collection of samples and the DNA extraction is done by Finnish government, so the ownership of the biobank material there's no question about that. It's public. So to summarize, we do FinnGen to use genetics to understand disease mechanisms, and it's powered by the unique Finnish population history, biobanks, and health data. So during the first 6 years, it was the GWAS-FIVAS era. We have more than 300 Finnish-specific hits on different diseases. Some of them have progressed to publications like these presented here. Like as you can see, there's a wide variety of different phenotypes we study. There is more than, we use the very well validated Finnish registry data from about 10 different registers for the formation of the phenotypes. The registers are used so that we cross-validate the information from different. So if you have a incident in a hospital discharge register, you need to have this kind of medication, and you need to have this kind of diagnosis here and there, etc. So this is a quite complex process, and it took a lot of validation in the first two 2 years about, but now it's working perfectly. We do analysis very automatically on Google Cloud twice a year, 4,000 endpoints against 20 million SNPs when imputed, and that's very well. I will come back to that. The program used for the cross-validation is called Endpointer. It's developed by my group, and I'm happy to share it with public access, so contact me if you're interested. So now we are past the GWAS-FIWAS, so everything against everything GWAS study, and what are we going to do now? So we have a lot of associations, and you know, having an association, it's just an association. You don't even usually know the variant, and I think it's the kind of a duty not to just go further and do more GWASs, but to go further into the analysis. What do these GWAS loci mean? And so we are looking for, and luckily I'm not the only one who thinks so, but this is the consensus of the steering committee and scientific committee of FinnGen, and we are now in FinnGen 3 have proceeded into the significance of identified genetic variants using biomarker analysis, and also by looking into, in a more conventional genetic epidemiology way, what influences disease progression. We have a luxury of having all the laboratory measurements from all the participants from the whole of Finland since 2014 because of our Kanta registry. We can get the medication changes, including hospital medications, from the registers and hospitals. We have uploaded clinical data observations from participating hospitals, and also naturally the health registers, more than 10 different registers that we can use for various things. So aim one is to deep dive into disease trajectory, and this is led by Professor Samuli Ripatti from FIM, Finnish Institute for Molecular Medicine, University of Helsinki, Finland. They're doing a great work on characterizing which genetic variants and which omics prevent and other issues, how your disease progresses. We have a longitudinal measurements from all of the participants, from the registers, starting from, say, 60s or even 50s for cancer register. Also, the laboratory measurements I mentioned, that we have often longitudinal measurements from those, and currently we're doing also longitudinal sampling for omics in a sub-cohort, in sub-cohorts. And then this is led by Mark Daly from Broad, is the characterization of variant association defined in FinnGen. Much of that work is actually being done in Finland by the Red Cross in Finland, etc. But anyway, the leader is our important partner from Broad. He used to be leading the FIM for I think 5 or 6 years, residing in Finland, but now he moved back to Broad. The aim one, we have had to select some of these like major disease groups, and we can't do everything, at least in the beginning. So we have selected pulmonology, IBD, kidney disease, metabolic diseases, eye diseases, rheumatic disease, and Alzheimer's disease, and Parkinson's disease, as you can read yourself from here. This is not only pharma decision, but this is voted by the steering committee about a year ago, to which we and any pharma had the same impact as any public actor. Study cohort is now the darker the blue is, the darker the more of the cohort comes from there. As you can see, most of it comes from Helsinki region or southern Finland, where also the population density is the highest, so it's not a surprise, and also it doesn't really matter because the southern Finland, the area you see in most dark blue, we have had a very heavy movement from countryside to southern Finland in the last 40 years. So we are like a mixture of people who live in Helsinki. For example, my father is from Kuopio in middle of Finland, and my father is from eastern Finland, from Kymenlaakso. So, but I myself, I'm listed here as somebody from Helsinki. Most of them are women, as always in our studies. They are more prone to participate. Thank you, and thank you, ladies. And at recruitment, the people are of early middle age. So this is now the distribution of where we got from. The THL Biobank and Arctic Biobank, we provided altogether about a little less than 200. This is the so-called legacy samples, and then most of these other 300,000 have been collected since 2017 when the project started, and that was where the government money was used for. As I said, we have health data based on longitudinal national health registers. Altogether, we have 500 million, half a billion, if you go Americans for a billion, data points for health. We have almost 1,000 average data points for individual. Remember, we follow them from registers which are pretty well characterized since 70s in Finland, so most registers were actionable already then. Then it's done on Thermo Fisher array and called FinnChip, and which is mostly their UK Biobank array, but enriched with Finnish specific variants and haplotype tagging SNPs and some pharmacogenetics. So we have lab data, as I said, we have clinical data, we have so-called deep diving study groups that we collect specific clinical data, say imaging data for Alzheimer's from clinics, and of course omics. Lifetime health data, as I've many times said, the thing I really wanted to say here is that the data curation is a major thing. We have in FIM the data analysis group who do the data curation, and they have done a great job in there. It's led by Mitja Kurki. There's more than 20 postdocs working just for the data curation and for the analysis, of course. I have a lot of numbers here. I'm not going to go through all of them, but you can see what kind of a data FinnGen has. So hundreds of thousands of different data points from different registers, and of course this is a resource, but it is also a big job to make sense of all of these, to actually dense this into a credible endpoint or a credible, say, disease progression or disease subgroup. At the beginning, we had even trouble differentiating diabetes type 1 and diabetes type 2 from each other, given the registry data, but now we are pretty happy with what we can do with that. So these are Finnish registers. You can see that there is a lot of them. We are pretty proud of the registry data in Finland. What we have, this is not FinnGen. This is I'm proud of as a Finn, not as a FinnGen person, representing this. This is something that the Finnish clinicians have been doing for decades, and the data is especially nowadays with the electronic health records, the clinicians cannot escape. They have to fill out these things they're mostly used for administrative use and clinical use, like but the secondary use is for science, and that's where we employ it. Omics data is done, as I said, on Thermo Fisher array. It's been working very well, and we're happy with the collaboration with Thermo Fisher. The transfer of the data. It's actually everything is done in San Diego. THL, my institute, sent the DNA there. The genotyping has been done. And we got the data back through Google Cloud. Now we're doing proteomics. We are leaning towards O-links, but we have done also some SonaScan. Mark Daly's project is doing some scRNA and ATAC-seq. We have legacy data for, and this is mostly from THL Biobank, for Nightingale and single cell painting as well for specifics projects. There's a lot of data. I will not go through this, but this is a wealth of data, and to describe how much the work is done, there's more than 1,000 registered scientists who have permission to analyze the data in Google Cloud. So the timeline is like this. So we have data freezes for FinnGen 3. We are now doing the data freeze 14. We have just received the data to THL. We're still doing this thing for the project. And there will still be one data freeze before, and of course we are planning continuation after 2027. We have some set milestones for omics as well. But it's not only a data repository source or infrastructure. We provide analysis and results tools and support. We have a research environment that serves well the scientists. It's quite easy to do. We have an online web course you have to pass before you get to analyze this for data security and also how to analyze this thing. But mostly everything, if you want to do GWAS, you can do it quite automatically there by selecting your data. It's even a guy like me can do analysis there. Of course, we do core analysis, so not everybody needs to do their own GWAS unless they have some specific, let's say, cohort they want to do it, but we do GWAS, FIWAS automatically, also again, because we update the registry data, so we get new cases, new mortalities, etc. We do fine mapping automatically. All this is done by algorithms. So nowadays the analysis group is mostly pressing enter and enjoying the fruits of their work. But then we do co-localization results of the GWAS hits. These all are, if you want to see this, go to results12.fingen.fi. It's free website. Everybody can see these aggregate results there, and then you do most of this stuff yourself. And I'll leave this. I'll just show you us here quickly. And thank you. But I'll show you, if you want, take a picture of this so you can use these QR codes to get to the tools and resources. And the aggregate data is available to anyone. I'll just say for the end that actually most of the publications mentioned in FinnGen nowadays come from China. They have been really enthusiastic about using our aggregate data. We are not sure whether we are happy about that or not. But anyway, it just describes how useful this data may be. Thank you.
Thank you very much, Markus. Now we are moving on to the last presentation of this session, and the last presentation comes from Professor Janis Klovins, very well known to you, all of you, and he used to be a director of the Latvian Biomedical Study Center. Now he's a chairman of the council and controls everything. So what is interesting, he's going to talk about common variants and rare variants, how to combine them together in diagnosis and prevention. The floor is yours.
Okay, thank you. And indeed, today I will outline the progress that we've been doing in establishing the genomic data information system in Latvia. That I'll devote first part of my presentation, and then move to some examples how we can use this resource both for the improvement of diagnostics and prevention studies. So first of all, I want to remind that this initiative, the establishment of genome data information system, is a part of the larger initiative of 1 million genomes, the European initiative that was established in 2018, when also Latvia became the member of this initiative. And as a core data set for this genomic data information system is the Latvian genome reference. And this is indeed a very successful project that has been halfway, at least from the sequencing point of view. First of all, we have created a population cohort and 3,500 individuals at the National Biobank. Then we have also performed the whole genome sequencing for all these individuals, and the integration of these sequences, clinical, epidemiological, and also some tools that has been produced to use this information is now underway, and we expect this data system to be ready for in production at June next year. So, and I have to also remind that this national initiative is strongly linked with two important projects in Europe funded by Digital Europe. First of all, this is the Genome of Europe project and also the European Genomic Data Infrastructure, GDI project, that together aims to create really the world-class European reference data resource, because this is something that we as Europe are really missing. And in the first step, the aim is to have at least 100,000 evenly distributed genomes across all the European countries, depending, of course, of the the density of the population in each country. So, a few words about the whole genome sequencing, what we have done. First of all, we used already established sample management workflow that we had in the biobank, starting from the blood collection, DNA isolation, the sequencing, also adding the data analysis, primary data analysis and transfer to the storage capacities. For the sequencing, we use both the short and long read technologies, and we aimed for the minimum coverage of 30x to really achieve good representation for each genome. For that, we actually had to do some resequencing. As you know, the read count really follows the distribution, normal distribution. So basically, there were some samples that we have to resequence. And we managed to establish this workflow for the sequencing that allowed us to use, to do at least 200 genomes per week. What are actually the people that are involved in this project? So the participant recruitment actually was conducted on a voluntary basis through the dedicated online platform that also allowed each participant to provide the informed consent, and importantly, including the agreement for secure data sharing within the European infrastructure. So even taking into account that this was self-selection, that we achieved a very good representation of people involved from different regions, as you can see, with some minor deviations. They almost represent the actual population density in each of the regions, which is in the red bars on the top graph. We also have different age groups, and of course, as already in the previous presentations, majority of the participants are women. From the self-reported ethnicity point of view, the majority indicated themselves as Latvians, but we also have other minorities, just also indicating there is inclusiveness of this project, and also that has to be, all the other minorities has to be represented in a genome pool of Latvian population. We developed the data analysis, standard data analysis workflow for analysis. We picked up the different file formats from the sequencing to be used for different purposes to store in our final storage system. And we also followed the initial recommendation of the Genome of Europe for the data quality that you can see on the pictures, achieving, of course, since we sequenced, we had the good mean coverage, but we also have a good evenness, low duplication rates, and other parameters of the sequencing quality criteria. So let's talk about this genome data system that we are creating. First of all, data collection. So as I said, the basic material comes from this genome reference. This is the core data set. But we also established this system so that all participants, all researchers, and also clinicians can deposit the genomic data into this system. So meaning that this will become, in time, the main genomic data storage system in Latvia. So far, we've been using the HPC at the Riga Technical University for the analysis, but the aim is to create a secure data environment. For that, of course, we need more servers for the actual calculation inside this facility. And importantly, but the most important part indeed is this portal, where we are creating different environments for different users. And importantly, also having this dynamic consent module that helps to manage participants' consents. Importantly, this is actually the place where this system is created is right here at the National Library, which is one of the secure data centers certified in Latvia. Let's now go through the different environments that we are implementing in this system. And the first one I would like to emphasize is really the environment for the participants of the project, participants whether they come from the research project or from the clinical part. So each person whose genome is deposited here in this system can access the portal through the authentication using electronic IDs, and can access their individual genomic data, and most importantly also there is a place to report the research results. Importantly, there's also this dynamic consent module that allows to withdraw or change the consent form. And there are also some, at least preliminary results that we can report to participants. In this case, we are planning to put there the polygenic risk scores for some of the verified diseases here, and also so that they be accessible for the individual level results to the participants. And importantly, the participants can also take opportunity to participate in different open projects that are created in this system. Next level is the working environment for the clinicians or other medical personnel. And importantly, as an aggregated data, without any specific authorization, there will be a possibility, and this will be the first functionality, to query the Latvian reference genome, like for the clinical geneticists, very important tool to look at the variant frequency and different annotations of variants that are characteristic for the Latvian population. But the next level, of course, is through the authorization from the individual patients. The doctors will be able to also look at the individual level data and use this information to support early prevention, the personalized therapy, and some pharmacogenetic decision making as well. Of course, this tool can be, this resource can also be used by researchers. The first important step is to really query this database and metadata to explore what information is there, what is available for the research. And then as the next level, each researcher, as soon as there is some approved research projects and ethics has been cleared, they can access the pseudonymized genomic and molecular data. At the first stage, we will not be able to ensure this safe working environment for the analysis, but the plan is to really have it also here, so the data are not transferred outside this system. And of course, very importantly, there is also a system that allows to create new studies, invite participants, and create some questionnaires for the following studies. Another functionality level is technical, so somebody has to put the genomic data in there. So we have created the data submission portal. We use the metadata definitions based on the EGA schemes, and metadata can be dynamically defined, knowing that there are different environments, different sequencing machines, and so on. So basically, this is flexible and there is possibility to change. Very importantly, we will use this data system to link with European Genomic Data Infrastructure, GDI. We already started to work on actual tools that are needed to adjust our data structure to the one which is used by the GDI. And importantly, one of the plans also is to use this information and environment to create a federated EGA, so for the open science, especially for the large data, for the sequencing or other molecular data to be used for the open science purposes. So one of the tools that we already created is a polygenic risk score calculator. At the moment, it accepts the whole genome sequencing data, which is not that often. And we have optimized several polygenic risk score models for the different diseases, specifically for the Latvian population, using previously genotyped samples. But importantly, this interface there's potential to be customized. So basically, somebody can put different polygenic risk score models for different diseases as a packs what they need for their research or probably clinical work. And so far, it is not integrated into the system, but we will use the data outputs from this risk score calculator to present them to the individual participants. So basically, this is one of the what you can see is how the actual individual data presentation for the polygenic risk score will look to the participant. There are several, now I'm moving to the several implementation examples and basically telling about how we used already sequenced cohorts to improve diagnostics in previously undiagnosed cohorts, which is more on diagnostic side, but also to show that these cohorts we already have in our hands is very useful to enable targeted participant recruitment for the actual prevention-oriented studies. So, and the whole genome sequencing actually has this unique, not unique, but quite clear capability that it can both detect Mendelian mutations, inherited mutations, and also you can calculate the polygenic risk score. So, and for example, in the genome database of Latvian population, we have several cohorts with the suspected inherited conditions, and importantly for those cohorts, there is no comprehensive genetic characterization performed at the clinical setting, so basically there is no ability to confirm diagnosis or guide treatments, and that's why we applied whole genome sequencing to these cohorts to address this gap. I will talk about the low-density lipoprotein C polygenic risk score, and already in previous talks you heard about the familial hypercholesterolemia as one of the cases, and also yesterday Professor Gustavs Latkovskis presented this study where indeed from the quite large cohorts from the registry we were able to finally identify, and in this case this is only a part of this cohort. From 300 people we identified 72 patients with pathogenic mutations. Some of the variants are unique to Latvian population. But most importantly, and what was mentioned already yesterday, these pathogenic variants explain only 21% of the FH phenotype, but applying and testing different polygenic risk score models, we actually found that at least the latest ones that if we choose the 95th percentile as a threshold for important finding, that allowed us to explain additional 40%, so totaling as at 62% of explained phenotype, which I think is a very important result. And indeed, as already was shown in Anders' presentation, that actually this clinical approach for the different type of genetics is different. So, and now this can be implemented into practice, both for follow-ups, treatment, and other needs from the clinician point of view. And finally, the one example where we really trying to use existing cohorts and also including this population reference cohort as a source for the studies that are directed for the prevention research. So this is the study what aims to really the targets metabolic people with metabolic syndrome or early prediabetic dysglycemia. This is the one criteria for the invitation. The second one, actually, what we wanted to do, we wanted to actually do the genome-guided selection of participants. So since all the genomes are sequenced, we actually can use this information. And aim of this study is to see whether the high or low polygenic risk score actually has influence on the effect of the diets that people can use for the to improve the conditions. In this case, to slow down or prevent dysglycemia. So this cohort goes through the different type of diets. And since this project was started earlier, we did not use our yet developing system, but used another tool for the recruitment developed by a very successful startup here in Latvia, Janis. And I really also encourage others to use this system, as it really provides a lot of advantages for the management of the patient cohorts, enrollment, and reporting back results as well. So the few preliminary findings that we found from this study, for example, first for whole involved and analyzed patients, what we see directly is that, for example, polygenic risk score strongly correlates with the age of onset for some metabolic traits. So example, on these pictures, you actually see only those participants that already have either increased weight or hypertension based on the existing therapy information. And in these sub-cohorts, what you see is actually when you compare different quartiles of polygenic risk score, the distribution you see, for example, with the first and fourth quartile, there's 8 years difference in the onset of these traits. So basically, this is something that really indicates that there is strong influence on, and then I should remind that this is only the type 2 diabetes polygenic risk score. Of course, we will also have the ability to look for the other polygenic risk scores in this study. Another interesting findings that can be also suitable for all of us, what we see is that what we checked, first we divided all this group of participants in 3 groups based on their physical activity level, and what we saw that actually the correlation between the polygenic risk score and either BMI or waist is only in the patient group that are not active, meaning that really even the moderate physical activity actually can mitigate this genetic predisposition, at least to these traits like related to adiposity. And the first findings from the actual diet study, there are still not large number of participants, but the first findings also show that polygenic risk score indeed influences the weight loss response. So the patients with lower polygenic risk scores actually experience greater weight loss, while the opposite for the patients with or the participants with a higher PRS value. We also have metabolic analysis performed on these cohorts, and we also see that some metabolic signatures are associated with the polygenic risk score as well. So the main conclusions from all this presentation is that indeed the Latvian genome reference already have some important information that can be delivered and additional cohorts that have been sequenced. And of course, this information can be used both for diagnostic and also the prevention studies. And I would really like to thank my group at the Latvian Biomedical Research and Studies Center for hard work that is done to produce all these results, also in preparation for different larger studies. And of course, my deepest gratitude is to the patients that actually and healthy people that participated in these studies. Without them, it would not be possible. I believe that among these participants, there can be somebody from the audience as well. For those that are waiting their results back, so I encourage to wait a little bit longer. So since we're still implementing this system, and we can deliver these results as soon as we can. Okay, thank you very much.
Thank you, Janis. I guess we still have some time for questions. So Markus, please move to the stage.
Let's start with the questions from the audience. If there is any question. Okay, yeah. There should be microphone. Okay, the microphone is coming.
Hello everyone, Audrona Sparte-Jansen, I'm representing Clinical Research Organization. I would like to ask you, do you have plans to promote Latvia genome in public for commercial clinical trials, like cooperating with European Medicine Agency and the portals?
I think this is certainly possible. Honestly, we don't have any clear strategy at the moment, at least when we are talking about the specific, like clinical, I don't know, drug-based clinical trials or something like that. And you also have to remember that at least in this smaller cohort where we have this best genomic information, these are mostly people that are very randomly distributed across different diseases. So basically, but if there would be an interest for that, and I think this is also the part of the work packages that are the Genome of Europe and GDI is working to have really make this case for the industry as a potential to use. So I think that will just come in with time.
Yeah, thank you. Are you cooperating with Latvian Rare Disease Association to collect some rare genomes?
The rare genomes actually come from the different studies, also from the clinics. I didn't have time to go deeper, but we have collected already, especially the cases for which we did not, there is no kind of positive diagnosis from the exome sequencing. This is the kind of major target so that we can do the whole genome to improve the situation there. Yeah.
Thank you very much. My name is Daniel Rivas. I appreciate a lot of the effort that you're putting into this work, collecting all this data. I'm wondering, because it was not clear, at least for me during the presentation, how open access are these, the information in these biobanks?
Maybe we can go through the different biobanks. Yeah.
If I start, in FinnGen, the data is open to all the participants. That is the 11 biobanks and their background organizations, which are the local universities, and then for pharma. The individual level data is not available outside the consortium. We are planning to do, well, there is different plans. I'm rooting for a plan that after 2027 we would be like UK Biobank, giving data to anyone, but I'm not the sole one making the decisions. There are people thinking otherwise as well. So let's see what happens after 27.
Estonian Biobank is also open for all researchers, so clear access rules are on the website, and the principle is that tools are coming to the data. Data are not moving outside.
Okay. For the Latvian Biobank, of course, this system will facilitate this access, but it's not ready yet. But as for the Biobank, we have calculated, and depending from the year, around 5 to 10% of all samples that we have, this is at the sample level, are actually required and used in the different projects. Since we have this, I would say rather, not very supportive legislation, at least for the National Biobank, this is often more difficult to provide samples and data outside the Latvia, but we do that. It just requires more attention and some additional paperwork to be done. But we hope that with this new biobank law that is now in the Parliament, that situation will improve.
I forgot to mention this, that all the genomic data and the omics data is available given the Finnish biobank law through access through various biobanks. But the sad thing about the biobank law is very good. But we have a law on secondary use of the data, which is then the phenotypes, and that doesn't allow us in FinnGen to share the phenotype data, the registry data, etc., to third parties. So we are restricted by law, but you can get access to all the data derived from the samples through Finnish biobanks. And if you are interested, there is a website called finnbb.fi where the access is provided.
Thank you very much for the information.
Hello, I'm Елизавета Sokolovska from Latvia, and I represent both research institution, but also I'm a practicing clinician, and therefore I have a question from the point of end user. The polygenic risk score, which you as a research organization, biobanks, utilize, how well it correlates with the genetic tests which are available on the private market of preventive medicine clinics, anti-age clinics, and so on. So how should we direct our patients?
I have to say that it correlates very well. And in our case, all biobank participants got the PRS, and via portal Minu geenivaramu, everybody can access the data, their own data. And when it was opened last May, first 6 weeks, I guess close to 100,000 people actually logged in and were watching, cardiovascular and pharmacogenomics and some other stuff. Also, how much coffee you can drink and when. So basically, I would say it correlates very well. And, it's, but, we have to do it just, we have to do it more.
Yeah, the patient downloads the data, and then he goes to some kind of doctor who understands something.
Patients own the data. They can download data from biobank, and they go to some, let's say we have a private hospital called Janis for 4 different cancers. They calculate PRS. This is one thing. This is clinical thing. But also from biobank, we deliver data to all participants, and they have to go into system. You enter like into the bank, you use your ID code and so on, and then you can see what is on you, what has been calculated. But this is not the medical information, strictly speaking, because universities are not providing health care. We are providing information.
One small comment to Professor Metspalu's comment is that, yes, the polygenic risk scores usually correlate very well if they are done, the background work has been done in the same ethnic group. And here we are, most of us are Westerners, and it would correlate well. But then if you move to another group, say from Western Europeans to Asians, you may have some trouble. Mostly the trouble is that the prediction power is less. It's not usually to the wrong direction, but the PRS is not working as well. But as Professor Metspalu said, usually they correlate pretty well across Western populations.
Okay, I can add to that. Of course, the question also relates to how reliable are they, even if they are super precise, how reliable are they for the use in the clinical practice? Then I think it's quite clear that it depends from the needs. It's not a diagnostic tool anyway. So basically, we have to remember that polygenic risk score is risk estimate. So basically, it is the same risk as the increased cholesterol or whatever. It should be looked as just one component of the many different components that can lead to some probably some change into the management. So, but of course that is for the doctors to decide. Yeah.
But still, if my patient is not in the biobank, and he asks me, doctor, so I want to determine this polygenic risk score. How do I know? How do I choose the clinic which provides? So what keywords should I look on to choose the good test, so to say, for my patient?
So basically, in our country, it works like this. You know, if there is someone who is not in a biobank, actually goes to this hospital, this private hospital who is providing, they are ordering this array test, they get the genotypes, calculate, impute, calculate PRS, and compare it against 200,000 plus people what we have, and see where this risk is for this particular person. So we had just early this week, there was a big story in our newspaper, somebody did like exactly what you are saying, and find out that is very high risk for prostate cancer. Immediately went to the hospital, they did MRI and discovered first stage prostate cancer. And this guy was very happy, so now I'm saving my life because first stage is much well treated, much better treated than third phase when you are already feeling it.
Okay, for example, in Latvia, actually now, at this moment, this polygenic risk score calculator is working. So basically, if you already have your own genome data, you can put it there. At the moment, there are only 5 diseases, but all of them are adjusted. And what you get actually is this projection on the Latvian population, which is important, I think. Of course, I don't think it will matter much to the other Western populations, but still, I think that this is one important thing to know. And then, yeah, I'm not really, I cannot provide a recommendation at which commercial facility you would be able to get the best genotyping.
That's an important question.
Yeah, that's another question, but it's as soon as you get these data, so you can actually even now use them here.
To be honest, I'm a clinician also. I do internal medicine, and currently the polygenic risk score is very at these baby steps. And the best science is done in the places where Professor Metspalu is working in the public side. The commercial providers, it's very hard to determine whether they are doing, because it's not just genotyping, it's algorithms, it's imputation, it's what background population they use. So I wouldn't put my money in commercial providers today. Maybe in future, but I can see it's very hard to evaluate. I have paid for a couple of these, and it's really hard to determine the science behind them because it's not like simple. It's quite simple, but it has its glitches.
Thank you very much.
Okay, if there is no more question, I think we also over the time, like 5 or even more minutes. I would like to thank all the presenters, and thank you.
Thank you.